Hi, I'm Tyler Farah, and I want to welcome you to this video about amino acids found in proteins. Amino acids are an incredibly important class of biological molecule with applications in biology, in general chemistry, organic chemistry, and biochemistry. So understanding certain key features of these molecules is critical to your success on the MCAT. In this video, we're going to learn to analyze what functional groups are commonly found on proteinogenic amino acids. We'll also analyze two naming conventions, the alpha convention for designating amino acids and the LD convention for designating isomers. Shown on screen are two examples of amino acids used in the construction of proteins. In fact, one of our targets today is to be able to identify proteinogenic amino acids. Meditate for a moment on the meaning of that word proteinogenic. You probably are familiar with the idea that there are 20 amino acids used by your ribosomes to make proteins. And yet it may surprise you to learn that there are over 400 known amino acids total. Clearly, to successfully answer a question like the one shown on screen, we'll need to recognize key characteristics of those molecules used for proteins versus those amino acids that are not. Let's begin our study by considering a very common proteinogenic amino acid, asparagine. Notice that all of asparagine's functional groups center around a central alpha carbon. That makes asparagine a chiral molecule, and in fact this is a feature shared by most proteinogenic amino acids. Now, of the functional groups, asparagine has both carboxyl group and amino group, and in fact this is where the terminology amino acid comes from. Now, if all amino acids have both amino and carboxyl group, where they differ is in the identity of their R group. Asparagine has an amide functional group as its R group. However, you'll also see acidic R groups, basic R groups, polar R groups, and nonpolar R groups. The variety of R groups really reflects the variety of proteinogenic amino acids overall. Finally, it's not often pointed out, but for completeness' sake, let's recognize that the fourth functional group is a hydrogen atom. You've heard that term alpha used several times in this video already, and it's a critical term for describing amino acids because all of the amino acids used for the construction of protein are alpha molecules. The term refers to the position of the amino group relative to the carboxyl group, and its origin is in IUPAC naming conventions. The highest priority functional group on the molecule asparagine is its carboxyl group, and the carbon that is bound directly to the carboxyl group is often termed the alpha carbon. Notice that the amino group is also bound to that alpha carbon, hence it is an alpha amino acid. Now, that is not to say that all amino acids are alpha. Consider by way of counterexample GABA. GABA has a carboxyl group and an amino group, so by definition it is an amino acid. And yet looking at the carbon backbone, we see an alpha carbon, the beta carbon, and finally the gamma carbon to which the amino group is bound, justifying the name gamma aminobutyric acid. This illustrates a very important point about amino acids in your body, and that is to say, some biologically active amino acids are not alpha amino acids. You may recognize GABA from its role as a neurotransmitter. It's only the 20 proteinogenic amino acids that are necessarily alpha amino acids, and this gives us a great criterion for recognizing which amino acids are proteinogenic and which ones are not. Now, let's revisit a point that was mentioned earlier on in this video, and that is that most proteinogenic amino acids are chiral molecules. Shown on screen are the two enantiomers of serine. What's interesting about enantiomers as regards amino acids is that biochemists have settled on a slightly different naming convention then is used for identifying mirror image molecules in most of the rest of organic chemistry. Specifically, you may notice that biochemists would refer to serine as either L or D serine. How do we arrive on this convention? Well, it's actually simpler than you might think. Simply draw the molecule in the configuration shown on screen with the carboxyl group at top and the R group at bottom. And then scrutinize the position of the amino group. If the amino group is on the left side of the molecule, well, then you're looking at the L isomer. And if it's on the right side of the molecule, that's the D enantiomer. The mnemonic for this is L is for left and D is the other one. It's still possible to describe amino acids using the old-fashioned RS terminology familiar from organic chemistry. So why come up with this brand new naming convention specifically for amino acids and other biomolecules? Well, there's actually a really good reason for that. You notice that some proteinogenic amino acids, like serine, are in the S configuration, while others, like cysteine, are in the R configuration. However, using the LD convention, all proteinogenic amino acids are L isomers, and that greatly simplifies the process of identifying proteinogenic amino acids. Finally, let's examine the amino acid glycine. You may recognize that glycine's R group is another hydrogen atom, and therefore glycine does not have 4 unique functional groups bound to its alpha carbon. That means glycine is not chiral, and that makes glycine unique because of the 20 proteinogenic amino acids, 19 are chiral molecules. Glycine is the only one that is not. And so now, let's return to conquer our study question from earlier in this video. Take a moment and answer this question now. Consider first answer choices A and C. On these molecules, the carboxyl group is too far separated from the amino group, and that means that these are not alpha amino acids and can be safely eliminated. Similarly, in answer choice D, we see that the amino group is on the wrong side of the molecule, making this in the D configuration, which also allows us to rule out answer choice D. That leaves answer choice B as an alpha amino acid in the L configuration and the correct answer. Let's now review all of these ideas using the amino acid threonine as our model organism, so to speak. All proteinogenic amino acids are going to have functional groups centered around a central alpha carbon, and those functional groups will consist of a carboxyl group, an amino group, and some sort of R group which designates the identity of the amino acid. All of these amino acids are alpha amino acids, indicating that the amino group and the carboxyl group are bound to the same carbon. And finally, 19 of the 20 amino acids are chiral amino acids. All 19 of those are the L isomer, and glycine is the only achiral amino acid because its R group is a second hydrogen atom. Thank you for watching this video, and I hope to work with you again real soon. Take care.